Hi everyone, welcome back to this YouTube channel. In today's presentation we are going to learn about the basic laboratory techniques using a microscope. This slide shows you an overview of the topics being discussed today. Let's begin firstly by discussing about wet mount slides. Through this video we are going to follow the step-by-step procedures on how to prepare for the mentioned slide techniques. The materials required for you to begin your examination is a microscope, microscopic slides, a cover slip, eyedropper, water, the specimen you wish to examine, and lastly some tissues. Obtain a clean microscope slide and a cover slip. A cover slip is very thin, permitting the objective lens to be lowered very close to the specimen. Place the specimen in the middle of the microscope slide. The specimen must be thin enough for light to pass through it. Using an eyedropper, place a drop of water on the specimen. Lower one edge of the cover slip so that it touches the side of the drop of water at a 45 degrees angle. The water will spread evenly along the edge of the cover slip. Slowly lower the cover slip over the specimen and water. Try not to trap any air bubbles under the cover slip. Air bubbles interfere with the view of the specimen. If air bubbles are present, gently tap the surface of the cover slip over the air bubble. Remove any excess water at the edge of the cover slip with a tissue. If the specimen begins to dry out, add a drop of water at the edge of the cover slip. Next, let's move forward towards the second microscopic technique, which is staining. This will also follow an explanation of the step-by-step procedure. The materials required for this technique is like the one mentioned earlier in this video. A microscope, microscopic slides, a cover slip, stain, eyedropper, water, the specimen you wish to examine, and lastly tissues. Obtain a clean microscope slide and cover slip, or clean it with the help of ethanol. Place the specimen in the middle of the microscope slide. Using an eyedropper, place a drop of water on the specimen. Place one edge of the cover slip so that it touches the side of the drop of water at a 45 degrees angle. After the water spreads along the edge of the cover slip, lower the cover slip over the specimen. Add a drop of stain at the edge of the cover slip. Clean the excess by placing tissue paper at the opposite edge of the cover slip. The paper causes the stain to be drawn under the cover slip and stain the cells. The stain used can vary depending on the specimen that you are examining. An example of a stain is methylene blue. The last technique being discussed in this video is heat fixation. This procedure also follows a similar pattern in comparison to the previous two techniques. The materials required for this technique are a microscope, microscopic slides, stain, a cover slip, an alcohol burner, lighter, tweezers used to lift the microscopic slide, the specimen you wish to examine, and lastly tissues. Obtain a clean microscope slide and cover slip, or clean it with the help of ethanol. Place the specimen on the slide. Light the alcohol burner with the help of a lighter, do not blow to extinguish, just put the lid back on. Lift the slide with the help of tweezers and then gently wave the slide across the fire. Then place the slide on your palm to control overheating of the specimen. Add a drop of the methylene blue stain, tilt the slide for the indicator to spread, and leave it for 2 minutes. Lift the slide and dip it in a beaker of water to remove any excess. This process is also referred to as destaining. Leave the slide until dry and lastly place the cover slip. Thank you for watching this video. Please do not forget to subscribe to this YouTube channel for similar videos.